Thank you for the introduction and the organizers for this opportunity to share our work. In this project, we are interested in how GCase deficiency modulate lysosome biology in iPSC-derived dopaminergic neurons. To do this, we first reprogram an iPSC line HT809 from a patient with both Gaucher disease and Parkinson's disease. This patient is homozygous for N370S, a rather mild mutation. Our plan is to generate a lockout outline to increase lipid accumulation and also correct it back to wild type to eliminate lipid accumulation. After reprogramming this line, we first sequence its whole genome. Its distribution of single nucleotide variants is comparable to the reference iPSC line COP2.1J. We also calculated its polygenic risk score for Parkinson's disease and Alzheimer's disease. Both of them fell within the expected population range. Together, HT09 has a rather neutral genetic background when GBA1 variants are excluded. To monitor iPSC differentiation into DA neurons, we inserted TD tomato, a red fluorescent protein, into the tyrosine hydroxylase locus. We also adopted a differentiation protocol that features biphasic activation of Wnt signaling. On day 35 of differentiation, over 70% of the differentiated cells express TD tomato. After replating, these cells stained positive for tyrosine hydroxylase, indicating TD tomato as a reliable reporter for TH expression. We also used RNA sequencing to compare iPSC and the differentiated cultures. Multiple DA neuron markers are enriched in differentiated cultures, and so are gene sets involved in neuronal development. To purify lysosomes from differentiated DA neurons, we inserted a lyso-IP tag into the cleavable safe harbor site in HT009. We used magnet beads to pull down lysosomes from the editor line as well as from the parental line as a control. Cross-referencing the human lysosome gene database, we detected over 100 lysosomal proteins in our dataset, and 83 of them are significantly enriched in the lyso-IP samples. That gave rise to an enrichment rate over 46% for lysosomal proteins as compared to 7% for non-lysosomal proteins. Therefore, our implementation of the lyso-IP workflow in HT809 is successful. After inserting these tools into HT809, we started to edit the GBA1 gene. As many of you know, GBA1 has a highly homologous pseudogene that is also expressed. Therefore, we think it is important to avoid off-target modification of the pseudogene while editing GBA1. After evaluating multiple guideline designs, we identified one that is efficient at targeting the gene but not the pseudogene. We used it to generate the lockout cell line. In HT809, N370S is the result of an A2G mutation, and GBA1 and the pseudogene shares identical sequences in this region, making it quite challenging to target one but not the other. 39 base pair downstream of this point mutation, the pseudogene lacks a 55 base pair sequence. Therefore, we used this region to target GBA1 specifically. We also built HDR donor vectors with selection markers to help with the enrichment of edited iPSC clones. Because we intended to correct both alleles at the same time, we added GFP to one donor vector and M-cherry to the other. These selection markers are also flanked with piggyback transposase regulation site to facilitate their removal. After the delivery of CRISPR machineries and the two donor vectors into the parental line, we use FACT sorting to enrich cells that are positive for both GFP and M-cherry. After growing up this double positive population, we deliver the piggyback transposase, then again use FACT sorting to enrich the double negative population. Sequencing this population, we successfully identified clones that has both allele reverted back to Y-type. After generating the lockout and the Y-type cell lines, we sequenced their whole genome as well. The parental line has a modest number of potentially deleterious variants, and our editing process did not introduce any additional ones. Also, it is great to see that the GBM variants are no longer present in the Y-type line. Because Illumina short-read sequencing has reduced the mapping quality at the GBA1 locus, we further use the PacBio long read to sequence the entire length of the gene and the pseudogene. Here shows the alignment for GBA1. When we look at the locale line, we can see sequence deletion in both alleles. When we look at the Y-type line, we can see both N370S reverting in both alleles as well. Besides these changes, there is no unintended modification to either GBA1 or to the pseudogene. After validating our lines using sequencing technique, we look at GCase protein level and activity. We first plotted GCase protein level in both iPSCs and in DA neurons. Overall, DA neurons have a higher GCase level than iPSCs. Very interestingly, N370S variant reduced GCase protein level in DA neurons but not in iPSCs. When GCase activity is measured using the FOAM-U assay, we saw higher GCase activity in DA neurons, and N370S reduced GCase activity in both cell types. We then performed GCase glycosylation analysis. In iPSCs, about 30% of the N370S GCase is sensitive to NOH treatment, indicating they are retained in the ER. This fraction is reduced to about 10% in DA neurons. Through immunofluorescence, we localized both Y-type and N370S GCase to lysosomes. After looking at GCase protein and activity, we used the mass spec workflow to measure its lipid substrate, glucosylceramide and glucosylsphingosine. GCase lockout dramatically increased the levels of glucosylceramide and glucosylsphingosine. On the other hand, N70S had a modest effect on glucosylceramide levels, but increased glucosylsphingosine level by more than tenfold. Therefore, we think glucosylsphingosine is a better biomarker for GCase deficiency in DA neurons. To look at cellular lipid homeostasis more broadly, we also analyzed our cells using a targeted lipidomic approach that measured over 1,000 lipid species. As you can, besides sphingolipids, you can see in local DA neurons, there is broad changes in many other lipid species as well. When we focused on sphingolipids, N370S had a modest increase in hexosylceramide and a concurrent decrease in ceramide. In the local cell line, there is greater fold change to hexosylceramide and the ceramide. After checking the lipid substrate, we purified lysosomes from the differential DA neurons. Comparing local lysosome to Y-type ones, GCase is the most reduced protein, followed by cathepsin F. In N370S, lysosomes cathepsin F is also greatly reduced. But GBA1 GCase is only reduced by 50%. There are 6 proteins that are significantly changed in both comparisons. So cathepsin F is reduced in both local and N370S lysosomes. Interestingly, GPNB is reduced in local lysosomes, but slightly increased in N370S lysosome. We followed up on GPNB because it is identified by Dr. Fatima's group as a biomarker in the set of samples from patients with neuropathic Gaucher disease. GPNB is also a PD risk gene, and the PD risk allele is associated with increased levels of transverse protein. GPNB is a transmembrane protein with a large luminal domain. We identified 2 antibodies that works well in our hands for Western blot. Both of them recognize the luminal domain of this protein. When we use the E4 antibody, it detects a single band. Its molecular weight corresponds to glycosylated full-length GPNB. Noticeably, GPNB level is higher in N370S, but lower in the local line. When we used the E1 antibody to block the same cell lysates, we detected B1 as well as 3 additional bands. Noticeably, B4 shares similar trend to B1, being higher in N370S, but lower in the local line. To investigate the specificity of these detected bands, we locked out GPNB in our GBA1 isogenic lines. As shown here, upon GPNB lockout, both B1 and B4 disappear completely, indicating they are derived from GPNB. On the other hand, B2 and B3 persisted in GPNB lockout lines, suggesting they are most likely non-specific signal. To further investigate GPNB expression and processing, we build 2 constructs. One is to express GPNB itself, and the other is to express a dual tag GPNB with a halo tag on its N terminus and M-state gold at its C terminus. When we transfected GPNB into H4 cells, we detect the B1 and the B4 again. This confirms that these 2 bands are indeed derived from GPNB itself. When halo GPNB M-state gold construct is transfected into H4 cells, we noticed molecular weight shift with both B1 and B4. This suggests that B4 represents an N-terminal fragment of GPNB that is now tagged by a halo tag. We quantified the ratio between B4 and the B1. We did not see a big difference between GBA1 Y-type and the GBA1 lockout cells. Therefore, we don't think GCase itself is involved in GPNB processing. When we use TMR ligand to label the halo tag, we noticed distinct localization between the halo tag and M-state gold. When lysosomes are stained with SR lysosome, the halo tag colocalized much better with SR lysosome than with M-state gold. Therefore, we think the N-terminal fragment of GPNB is enriched in the lysosome after cleavage. To see whether the lysosomal changes we see are driven by transcriptional regulation, we generated RNA sequencing data from these cells as well. N370S and the lockout DA neurons exhibited similar changes. When we look at a GPNB, it's a homolog PMO, and their mass regulator MITIF and the TFEP, we saw a clear reduction in the lockout cell line. We also look at a cathepsin F as well as other lysosomal hydrolases. Cathepsin F turns out to be the most reduced one in both lockout and N370S lines. Therefore, we think the transcriptional regulations definitely contributed to the lysosomal changes in these cells. To investigate GPNB level in patient samples, we reanalyzed the PPMI CSF dataset. In this dataset, GPNB level are measured by four probes. 3 of them recognize the luminal domain of this protein, and 1 recognize the cytosolic domain. Here, we stratified the participants based on their status for Parkinson's disease and the GBA1 N13S variant. The hetero refers to N370S carriers. The PD homo group include 4 PD cases who are also homozygotes for N13S. The first 2 probes reported higher GPNB levels in the PD wild-type group as compared to the control group. These 2 probes also suggested a further increase in the PD homo group. Therefore, we think GPNB has the potential to serve a biomarker to reflect lipid storage in GDPD cases. To summarize our work, we have generated a panel of GBA1 isogenic iPSC lines that is now providing a platform for drug discovery targeting GBA1 and also facilitating CRISPR screening to identify GCase modifiers. We also characterize lysosomal perturbations in these cells. Our data suggest the luminal domain of GPNB are likely processed into multiple fragments that are enriched in lysosomes. Also, GPNB may have the potential to reflect lipid storage in GDPD cases. With that, I would like to thank all the members in Ellen's group, particularly the students who work with me on this project. I also want to thank all our wonderful collaborators as well as